Okay, hi everybody. Great to have you here. As is often the case, we are ready to start a new chapter. So again, let me begin by sharing the PowerPoint. This is chapter 6, Introduction to Viruses, Viroids, and Prions. This chapter starts with a fascinating case study at the very beginning of your chapter and culminating at the very end. There's that kind of a two-part to this case study, but they introduce it there in the very first part of chapter 6. I'd like you to read that over. It's fascinating and delves into some really interesting biology with regard to the use of phage, which are special types of viruses that invade bacteria. We're going to be talking about that coming up in this chapter and in some subsequent chapters. So again, check it out. Case study, beginning of chapter 6. Well, what has pretty much governed our lives in the last year now is, of course, the SARS-CoV-2 virus. And you've probably all seen sketches or diagrams of the virus. These aren't actual photographs. These are animations oftentimes, or sketches of what the virus sort of looks like, falsely colored here. And we'll be getting into more of this as we get into the chapter, but in essence, these red structures that are sticking out from the surface of the virus, and there's also some smaller, less obvious structures here shown in orange and in yellow. These represent proteins. Oftentimes they're glycoproteins, at least those here in red. And what they do is they provide signature to the virus, and it's with these glycoprotein spikes, these big guys, that the virus uses to dock, if you will, with a host cell. The host cell has basically a receptor on its surface. If you think back to plasma membranes, phospholipid bilayer with embedded proteins, right? Well, some of those proteins regulate what enters and leaves the cell and all that good sort of stuff, but other proteins act as receptor sites that the virus glycoprotein spikes will literally dock with or merge with, and that's what basically is like a key unlocking a lock and you walk through the door, right? That's how the virus gets into the cell. It uses its special specificity with respect to the glycoprotein spikes and the host cell receptors, and the host cell has no alternative but to allow the virus in. Very interesting. Well, if we go back in history, we can thank our friend Louis Pasteur, who we talked quite a bit about in chapter 1. Remember, he was the fella that saved the French wine industry. He put to rest the theory of abiogenesis and spontaneous generation, right? Well, he didn't stop. He didn't rest on his laurels. He actually was one of the first to hypothesize the presence of something smaller than even bacteria, which turns out to be a virus. In fact, he was working with rabies virus. He actually hypothesized that these particles existed. He didn't know what they really were, but he knew they weren't bacteria-like. They had very interesting different properties from bacteria. About the same time, at the turn of the 20th century there, two independent researchers were looking at plant diseases, and specifically they were working on what caused plant leaves to go from their normal nice bright green color to this sort of mottled yellow and orange. This is due oftentimes to viruses. This happens to be what's called the tobacco mosaic virus. They were working on tobacco plants, obviously, and they were the first, at least this fella here was the first to coin the term virus, meaning poison in Latin. Fast forward 60 plus years, and biology begins to spend much more time researching these entities and what they are and how they work. So virology as a formal branch of biology sort of came on its own in the early 50s and continues to be a very important branch, as you might guess, of biology with respect to its pathogenicity. Many types of viruses cause lots of diseases, as we know. So what's really interesting about these things called viruses is that they are acellular or non-cellular. They don't have a nucleus. They don't have the typical organelles that we associate, for example, with eukaryotic cells. They don't have a cell membrane. So they don't share many of the general life characteristics that we might ascribe to, say, prokaryotic cells or eukaryotic cells. One of the chapters that we already covered, I think, talked a little bit about some properties of life. And so these viruses just don't exhibit most of those. In terms of when scientists think viruses first developed, we don't really know. We don't have any sort of fossil record that we can go back into and look at like we do for eukaryotic and prokaryotic cells. But because we know that viruses require a cell to invade in order to replicate themselves using the cell's own building block molecules and other organelles, we are pretty convinced that viruses didn't exist until cells first evolved. Because again, viruses need cells to complete their life cycle. And we know, of course, that we can trace prokaryotes how far back? 3.5 to 4 billion years ago, right? And then eukaryotes came about 1.5 million or 2 million years after that. So when we look at viruses today, we already have established that sometimes viruses can target plant cells, like the tobacco mosaic virus that we talked about a few moments ago. We know that COVID-19 infects our cells. And you heard me talk about phage earlier with regard to their use in the treatment of this very sick scientist in the case study at the beginning of chapter 6. Phage is a particular type of virus that targets bacteria. So when we talk about a host cell that a virus needs, that host cell could be a bacterial cell, a prokaryotic cell. That cell could be a plant cell. That cell could be a fungal cell. That cell could be an animal cell. That cell could be a protozoan or an algal cell. So there are different viruses that target a whole range of different kinds of cells. I don't think there's any cell type out there that isn't potentially impacted by viruses. And that makes perfect sense if you think again about how they have come to kind of the evolutionary pathway of viruses has been intimately connected to all these cells. One of the earlier editions of your textbook in this chapter had a little blurb about viruses in the oceans, and that's why I just kept this slide. But your text doesn't really say too much about it, and I wish they had, because it's kind of interesting. I didn't realize that in terms of unnamed and unresearched, undiscovered viruses, if I was a virologist, I would probably go to the oceans to study the myriad of viruses that exist there that have never been studied. You know nothing about them. So that's certainly an unknown environment that we could certainly spend time exploring. It's anywhere you can go on Earth are going to be viruses, of course, anywhere you have cells. Let's read over that factoid. Okay, we've already said that in order for a virus to complete its life cycle, it has to get into a cell. And so we often use the term obligate intracellular parasite when describing a virus. Obligate simply meaning that the virus is obligated to get into a host cell before it has any chance of making more copies of itself, if you will. Now, it's the host cell, it's within the cell itself, intracellular, where this transformation occurs of the host cell, where now it receives new instructions to create more viral particles. We'll talk about how that happens coming up in this chapter. I am not going to go over all of these various properties of viruses, because by the end of the chapter, you can go back to this table and look at those bulleted statements, and they're going to make perfect sense to you. You're going to have heard about all of this. So again, I'm not going to spend time going through each one of these, but I would suggest at some point in time going back and revisiting this table if you want to get some general characteristics. It's a great, great table to look at. Well, we have talked a lot about sizes of eukaryotic cells and prokaryotic cells, right? We've defined, what a millimeter is, what a micrometer is, what a nanometer is. So I'm not going to review all that, but I think again, it's worth trying to wrap our heads around how small these viruses are, say compared to your typical prokaryotic cell. And that's sort of what this figure is doing. So we've got some large cells, at least they look large compared to the viruses, don't they? Here's a bacillus-shaped cell, our friend E. coli. Here's streptococcus, although you wouldn't know by looking at the single cell, but there would be a bunch of these linked together, right? We talked about that in chapter 4, I think, or 3, I guess it was. But nonetheless, coccus meaning spherical. We did talk about rickettsias. You read over the different types of rickettsias, which were really small, rather unique prokaryotic cells. And so these things look monumentous, don't they, compared to our viruses, which begin here with number 1 and go all the way to number 10. And of course, you can reference the table there just to get an idea of what some of these might be. And some of these look familiar to you, I would hope. Pox virus, you've heard of chickenpox and cowpox. That's kind of what a cowpox or a chickenpox virus might look like. Herpes simplex, that can cause cold sores. Here's our rabies virus that good old Louis worked on. HIV causes AIDS. Here's a flu virus. This looks somewhat familiar to you perhaps because flu viruses are coronaviruses, which also would include our friend COVID-19. This is a bacteriophage. It's a special type of virus that targets bacteria. And then finally, number 11, they just threw that in here as a protein molecule, hemoglobin. You remember from A&P that is a pigment found in our RBCs, our red blood cells, to which oxygen and to a lesser extent CO2 binds. So again, we're talking nanometers here in terms of size, ranging from fairly large viruses like megavirus, mega meaning big, of course, down to really, really small, 22 nanometers in length. That is extremely small. You would need an electron microscope, wouldn't you, basically see any of these viruses. This is about the cutoff where the compound light microscope could no longer be used, right? We'd have to use an electron microscope, and we do. So what does a virus have in it? What is it made of? Well, it's a very simplistic structure. It has evolved in a way that it doesn't need to carry much excess baggage with it, because who's going to do all the work in terms of making more viruses? Well, it's the host cell, right? The virus is going to get into. So the virus has the luxury of not having to pack a lot of stuff in it, because it's going to rely on the host cell to provide the needs to make more viral particles, because that's the goal, right? A virus's goal is to get into a host cell, and that host cell to make thousands and thousands and thousands of viral particles, which can then leave and go in and infect other cells, and the cycle continues. So let's talk a little bit about what is a virus composed of. And as has the last two chapters with regard to surveying the eukaryotic and prokaryotic cells, the book is using a similar kind of dichotomous key, if you will, breaking this down into categories with the outer covering and the central core region of the virus. And we're going to, of course, go through each of these and talk a little bit about what it's made of. As you can see, there's not a whole lot there, is there? Basically, what we have when we talk about a virus is an outer protein coat with an inner nucleic acid core. That, in a nutshell, is what a virus is made of. Outer protein, inner nucleic acid, period. Now, does it get complicated? Yeah, as we're going to see in a few minutes, there are different kinds of protein coats, if you will. There are different kinds of nucleic acids. When we say nucleic acid, we're talking DNA and RNA. Hope you know that, right? So let's start off with the outer covering and talk a little bit about this outer protein coat called a capsid. A capsid. So in this particular diagram, we can see the nucleic acid in sort of red purple, dark pink. And then in blue, of course, is the capsid, the outer covering, which in this particular virus has a bit of a geometric sort of property to it, doesn't it? And some viruses are very geometric. They're actually very beautiful to look at. Others don't share a crystalline sort of appearance or geometric appearance, but this particular one does. And so when we take into account the outer capsid protein and the inner nucleic acid core, we would call that structure collectively a nucleocapsid. So nucleo refers to nucleic acid, capsid refers to the outer protein coat. Now, this particular one, of course, has another term in front of it called naked, meaning that's all you got with respect to the virus. There's no outer envelope. It's naked. Okay, going back to the capsid for just a moment, we can see again how this has a bit of a geometric, almost crystalline morphology or architecture. So each of these little sort of pyramidal shaped components, right? And you see that there are dozens of them. These are called capsomers. So there's a capsomer, there's a capsomer, there's a capsomer, there's a capsomer. In this case, it'd be sort of like the various outer edges, if you will, of the naked nucleocapsid virus. But we know that in some viruses, we have an external covering called an envelope. Not all of them have it, right? There are a lot of naked viruses that lack the outer envelope, but here's a diagram of a cutaway view of an enveloped virus. And you can see, of course, the inner nucleocapsid, which does not look dissimilar to our naked nucleocapsid up here, does it? But external to that is this outer envelope. And you should recognize what these guys are. Of course, it's labeled. But remember back to our COVID-19 diagram we looked at earlier in the lecture, those particular spikes on the surface. Those are glycoproteins, which give that particular envelope virus a specific ability to target, again, certain types of receptors on the surface of host cell membranes. Now, what's really fascinating is how does an enveloped virus acquire its envelope? Well, we're going to talk about that, and I give you a little sneak preview here in red. But it's basically obtained from the host cell as the virus leaves. So more on that coming up. Okay, let's talk about the capsids, those kind of building block components that we talked about a moment ago that are part of the capsid itself. These capsomers are the, again, building blocks We talked about those not too long ago, right? Those were these kind of triangular shaped or pyramidal shaped structures of this particular diagram. But here we've got some capsomers that look very different. They're still the building blocks of the capsid. Okay, that doesn't change by definition. But in this particular set of sketches, we see these various capsomers making up a helical structure within which we see the nucleic acid being wrapped. So this is the formation of what's called a helical nucleocapsid. Is this naked or enveloped? If you said naked, you were right. There's nothing external to this. At least I don't see anything here, right? And in this particular electron micrograph, normally I try to give you the magnification of that, and I guess I did not do that, so I apologize for that. And I'm not sure if this particular diagram or photograph is even in your book, but this would probably be hundreds of thousands of times magnified. Certainly, looks to be from a electron microscope. But these long cylindrical structures here would be these. If you could look even closer, they'd be these sort of helical capsomers composing a helical nucleocapsid. And in the very core of each one of these would be a nucleic acid, either DNA or RNA. And here we can see sort of a cutaway view if we were to look into one of those viruses. Oh, here's our friend tobacco mosaic virus. Remember, that's what caused the tobacco leaf to turn kind of yellow-orange, right? Yeah, that's what causes that. Gets into the leaves, gets into the cells of the leaves, and that is the effect. And it hurts the plant, it could kill the plant. Okay. Here we've got, again, a helical capsid. In this cutaway view of this virus, we can sort of see that here. It's very helical. Only this particular helical nucleocapsid has an outer envelope, right? As compared to the naked helical nucleocapsid like we had with tobacco mosaic virus. Here we've got viruses like influenza virus, measles viruses, are enveloped helical nucleocapsids. Again, the helical nucleocapsid is shown here in sort of pink. Here it is in kind of red and yellow. And in the very center of that would be the nucleic acid. And then, of course, here's our outer envelope with the various glycoprotein spikes shown here in yellow and green. So helical is a real common capsid shape, if you will. The other common structural type of capsids are the icosahedral forms. Now, these are the ones that have the more geometric, crystalline, sort of cool look to them, I think. Kind of neat looking. So what we've got here, again, is a series of capsomers that make up the outer capsid. Okay, so this guy is right here, goes here, and there's a bunch of them all over the place, right? And then in the very core, which we can kind of see here in red, is our nucleic acid. Here's a 3D model of what an icosahedral virus might look like. Pretty interesting looking, isn't it? You can see these they call them fibers. They're not so much glycoproteins, although they may share similar chemistry to glycoproteins. I'm not quite sure. And then this is an actual highly magnified stain. Again, your book does not give the magnification, but it's got to be 600,000 times or more, I would think. And what's interesting is you can kind of see, can't you, the individual capsomers here. That's kind of cool. It's hard to pick out the pyramidal shaped components here because it's just hard to see the three-dimensional aspect of that virus, but that's in essence what it is. But what is, of course, noticeable are these very nice fibers sticking out, some of which have broken off here. Kind of cool. Now, this negative stain, you might remember from our discussion of staining, or we're getting into this in lab, I think this week actually. In a negative stain, the background is colored or stained, in this case black, and the object of interest, whether it's a bacterium or a virus, is white. A positive stain would be where you have a clear background or a white background, and the object of interest would have a color to it, purple or red or green or whatever kind of dye would be used. Okay, so let's go back to envelope viruses and talk a little bit about them. Most all animal viruses would be of this type, enveloped. And as I mentioned a little bit earlier, the virus acquires its outer envelope typically when it leaves the host cell. Now, there are some cases, as the second bullet here indicates, that are assembled within the cell, and we'll get into that process a little bit later. So they don't get put on the outer cell membrane, certain kinds of viral proteins, and then as the virus leaves, it becomes coated with the viral proteins. Some of these might be assembled within the cell during the time in which these various capsids are being constructed and the DNA and the RNA are being replicated. So more on that coming up in a few minutes. We've talked about the spikes before. They have special names, they have special numbers. A few years ago, the I think it was I forget now, H2 or H2N2 virus was real common. There's a whole host of different permutations of this. What these letters and numbers refer to, H stands for hemagglutinin, which would be in this case the yellow spikes, and neuraminidase is actually an enzyme. I know that because it ends in ASE. Those are the yellow spikes. And so what virologists will do in an attempt to differentiate different types of envelope viruses like this particular family, they use different numbers, and I can't tell you specifically if this has to do with when they were discovered, what sequence they were discovered, or exactly how the numbers are determined. But there's just like H1N1, there's H2N2, there's H3N3, there's H5N2. There's all a bunch of different combinations that basically say we have unique surface spikes, right? Specific hemagglutinin and neuraminidase spikes that categorize that specific named virus. Another function of the outer envelope, or in the case of a naked nucleocapsid, it's basically providing support, isn't it, as you might expect, or protection for the underlying nucleic acid. It's sort of like a coat on the surface. Now, in the case of an envelope virus, it sort of has the outer capsid, but it has an additional envelope, right? So it has kind of like two barriers, if you will, in terms of protecting that nucleic acid core. And sometimes it's really important to be able to do that because, as it indicates here, in the case of some intestinal viruses, they've got to get past the low pH of the stomach. If you ingest them, you might remember from A&P too, that the pH of the stomach is about 2. That's extremely acidic, right? And yet viruses can survive their trip through the stomach due to that outer protective capsid and or envelope that they might possess. And as I've mentioned repeatedly, another important function of the outer capsid and specifically the outer spikes characteristic of envelope viruses is their ability to dock with special surface proteins of host cells. And then, of course, the host cell has no alternative but to allow the virus in. Okay, let's talk about some sort of atypical, unique viruses. These are referred to as the complex viruses. We'll talk about two special groups of complex viruses. You see one of them here called Poxviruses. They're just very unique, strange, if you will. When you look at their outer structure, it's not your typical envelope virus or naked icosahedral virus. It has some very strange shapes. And so when you think of pox viruses, think of chickenpox, which I suspect many of us had as kids. Smallpox, which is a very nasty disease that hopefully we've eradicated from Earth. That's one thing that humans have been able to do successfully, is pretty much eradicate smallpox virus. Although our government and many other governments throughout the world are harboring smallpox in our government labs. And you might wonder, why would we want to do that? Well, it's called germ warfare. If it ever came to a point at which a country needed to use a biological weapon, this would be a weapon one could use knowing there is absolutely no protection out there in the general population, because smallpox has not been an issue here on the globe for a long, long time. So it would be devastating to a population if they were exposed to smallpox. Now, it's just sort of sad to think about the fact that we keep these potential pathogenic bacteria and viruses for that purpose. And hopefully it will never come to that. But I'm here to tell you that the US and China and Russia and perhaps other countries too have these sorts of microbes locked up, hopefully, and have access to if they ever need them. It's just kind of interesting, kind of scary, but that's the truth. So back to our pox viruses, let's think of a less nasty scenario, chickenpox, right? We've all had that as a kid, as I said, it belongs to this group of viruses that has a very strange outer layer of lipoproteins and these fibrils that stick out from the surface. You don't have the typical capsid made up of the capsomers like we saw earlier. This is a kind of a cutaway view or internal view of a pox virus. It is very strange looking things. And then the other special group that I've made mention of already are the bacteriophages, or sometimes we just use the term phage for short. This was again addressed in the clinical study at the beginning of the chapter. So bacteriophages, or phage, has an interesting polyhedral nucleocapsid, which you can see here. And then it has a collar and a sheath and these bizarre looking tail fibers that stick out. It's almost very sci-fi, isn't it? And you can see it in the middle here, this base plate with these pointy tail pins. Gosh, that's crazy looking. But the reality is, here's an electron micrograph of a bacteriophage, and we can see the exact structures. In fact, of course, the sketch came from the photograph. So you can see the sheath. Here's the capsid head. Inside of this would be the nucleic acid, either DNA or RNA. Here we can see the tail fibers sticking out, sort of like legs of a spider almost. Here's the tail pins. Yeah, it's crazy. The sheath. These are viruses that, remember, are targeting bacteria. So the prefix bacterial might help you remember that. We'll be talking more about their biology and how they work a little bit later. So in review, here's a nice figure 6.10 in your book, that looks at, again, those special groups of complex viruses, the pox and bacteriophage categories. And then this reviews, of course, the envelope viruses, whether they're helical or icosahedral, and then some non-enveloped naked viruses, which could also be icosahedral or helical, just lacking the outer envelope, right? And this just sort of breaks down those various categories and gives you some examples I'm not going to be expecting you to connect a specific type of virus in terms of what it actually is made up of. So don't freak out about this. There may be some that I might ask you about. For example, COVID-19, you know that's an enveloped virus, right? You know, tobacco mosaic virus is a naked virus, naked helical virus. So if I make mention of several, then I think you should know those, but do not worry about memorizing this particular box. Okay, we've talked about the outer covering. Now let's get into the central core of the viruses and talk a little bit about what's there. And we said earlier that for the most part, we're talking about nucleic acid, right? That being either DNA or RNA. So we can talk about DNA viruses or we can talk about RNA viruses. You don't have a virus that has both together in its core. It's either one or the other. And what's rather interesting, but not surprising really, is the fact that when you look at the genome of the nucleic acid core of a virus, and by genome we're talking about what genes are there, that we're talking about a rather small number of genes. They say anywhere from 9 to as many as 2,000. That's a relatively low number when you compare how many, say, E. coli has, or how many your skin cell or nerve cell or muscle cell or brain cell has, right? And the reason for that is because it carries with it, remember, a small instruction booklet. Because who's doing most of the work? I said earlier, it's the host cell, right? Be that a bacterial cell in the case of bacteriophage, or your cells in the case of COVID-19. It's the host cells that do all the work. They have all the cellular machinery to make more virus. All the virus has to do, for the most part, is provide the small little instruction booklet to the cell, and then the cell reads this and makes more virus. And that instruction booklet is basically the genome of the virus. Okay, and that's basically what the second bullet kind of talks about. Okay, you heard me talk about DNA viruses and RNA viruses. Well, we're going to complicate things just a little bit. Up to this point in time, when we've talked about DNA, think back to ANP1, chapter 3, I think it was, 2 and 3. We have always said that DNA is made up of a double helix. You remember this, I hope. Made up of nucleotides. The nucleotide is composed of 3 parts: phosphate group, sugar, deoxyribose sugar, and a base. And the base is either adenine, thymine, cytosine, or guanine, right? And in the very center of that double helix is where the bases interact with one another via hydrogen bonds between the bases. If we have an A over here, what do we have over here? T, right? Hydrogen bonds hold those together. If we have a cytosine in this nucleotide, opposite it, guanine. It's called complementary base pairing. You should know this. So double helix describes the shape of DNA. That's what we've always talked about. Well, believe it or not, there is such a thing as single-stranded DNA. I'm not going to get into it beyond simply telling you that some viruses contain single-stranded DNA. And the group that I'm mentioning here, of course, is a group called the parvoviruses, or parvo for short. If you have a puppy or a dog at home, you've probably heard of parvo. It's a very nasty, dangerous group of viruses that kill lots of dogs every year. And again, I'm not going to get into specifics, but I do mention here the 2 kind of types of parvoviruses, some of which can target the digestive tract, others cause heart issues with regard to the dog. SS single-stranded DNA, DS double-stranded DNA. We've also said up to this point in time that when we think of RNA, be it messenger or ribosomal or transfer, those are the three types of RNA. Chapter 3, Chapter 4, ANP1. Go back and review that if you need to do that. We've always alluded to the fact, at least with regard to, say, messenger RNA, because we talk more about that, made up of sugar phosphate groups, right? The sugar, however, is called a ribose sugar, not deoxyribose like we have in DNA. And then we have those bases connected to the sugar. And what are the four bases of RNA? Do you remember? Cytosine, guanine, adenine, and in place of thymine, uracil. Remember that? Yeah. And we've always talked about RNA being single-stranded. Well, as you can see here, we also have this other type called double-stranded RNA. Again, I won't say anything more about it. Your book makes brief mention of a group of viruses called the retroviruses that are composed in their core of double-stranded RNA. So if you were to take a higher level, virology class, you'd learn all about single-stranded DNA and double-stranded RNA. But we're not going to say much more about it other than the fact that they do exist. Okay. Now, once in a while, in addition to carrying the nucleic acid, right, that all viruses have to have, some viruses will also package in them certain types of enzymes that are going to be required to help that virus undergo replication within the host cell. And this particular slide points out three groups of enzymes. And how do I know these are enzymes? Because it says there. No, how do you know? Well, I've underlined the suffixes, right? Again, ANP1, we talked about this. Anything that ends in ASE is an enzyme. Amylase, lipase, sucrase, peptidase, decarboxylase. Talked about those in ANP2 actually in the digestive system. So these are enzymes. And again, we'll review some of the functions of these enzymes later on, but when I say polymerase enzymes, I hope you remember from ANP1 that this group of enzymes is absolutely critical in the synthesis of DNA and RNA. There's DNA polymerases, there's RNA polymerases. Replicases, we never talked much about replicases, but this is an important group of enzymes that the host cell is going to use to help make copies of RNA. And if you make extra copies of messenger RNA, for example, you can make lots of protein down the road, because remember, RNA has got the instruction booklet to help the cell make protein in a process called translation. It also helps to have transfer RNA and ribosomal RNA working together as well, right, to help make the protein. But replicases help to replicate the RNA. And this particular specific enzyme called reverse transcriptase, we'll talk more about it a little bit later, but it's a really interesting enzyme that retroviruses like HIV/AIDS uses. HIV/AIDS is an RNA virus, and what it does is it uses this enzyme that it brings into the host cell, okay, and it reverses transcription. That's where it gets the name reverse transcriptase. Well, again, you should know what transcription is. If you don't remember, go back into your ANP1 notes and review that. We'll review it a little bit later, but you should go back and know what protein synthesis is and how it occurs in the eukaryotic cell. That would be highly recommended for you to do. Go back and review that. So it takes the RNA and makes DNA from it. It reverses the transcription process. And this is really crazy. The fact that some viruses, before they leave their host cell, okay, so remember, viruses get in, host cell makes new viral particles, and those viral particles actually steal some of the host cells' ribosomes and some of the host cells' transfer RNA molecules. Why would you want to do that? Well, because if you remember again from A and P that the ribosomes are the eukaryotic organelles where proteins are assembled. What kind of proteins do we want to make? Viral proteins. We want to make capsids, right? We want to make more viral particles. We need viral capsids. And those proteins can be assembled and are assembled by ribosomes. So that's just kind of crazy that these arenaviruses actually will steal some of the host cell organelles to help down the road replicate. That's crazy. Okay, so how do we classify and name viruses? Now remember, these are acellular, they're not living, but yet we do have a means to classify and a taxonomic nomenclature that we use to identify different kinds of viruses. So we look at all sorts of things. Virologists look at the shape of the virus. Is it a DNA? Is it an RNA virus? It looks at the genome inside. There's a whole host of characteristics that would be assessed in helping to determine how to classify a group of viruses. The family names, notice, are italicized and end in the suffix viridae. So here's an example, herpes viridae. Well, that of course contains the herpes viruses. I hope you remember that, or would conclude that via logic, right? So herpes viridae, if you ever see that word, that's a family. That's a more overarching taxonomic category. And then as we go down to more specific taxa, we encounter genus. This should sound familiar to you. You know, our genus is Homo. Escherichia coli. Escherichia is the genus of that bacterium. Well, viruses are also assigned a genus name. And those are likewise italicized and end in the word virus. So herpes simplex virus, that would be, simplex virus would be the genus. There's a whole host of simplex viruses. Herpes is just one of them. But in our everyday conversation, or when we read about this in the newspaper, or we hear about it on the news every night, you're not going to see these family names or genus names. This is, you know, really more confined to academic literature and that kind of thing. But we would use common names, you know, AIDS virus, COVID-19, rabies virus, parvovirus, herpes simplex 1, herpes simplex 2. You've all heard of those. So those would be the more common terms we would use when we talk about viruses, because most of us are not virologists. With respect to pathogenicity in humans, there is a table in your book, table 6.2, that break this down into DNA and RNA viruses. You can see again the family names all end in viridae, right, as we promised, and the genus all ending in the word virus, as promised. Here's our vernacular common name. Some of these look familiar to you, I hope. Herpes simplex 1 and 2. Human papillomavirus, HPV for short, right? You've heard about those probably, and how we can now inoculate our sons and daughters when they're 10, 11, before they're sexually active, to eliminate the possibility of cancer down the road. Here's parvovirus. Polio virus, you've heard of polio maybe. Hepatitis A. Norwalk virus, this is the stuff that often gets into cruise ships and people get sick. You know, thousands of people on a cruise ship have to go back to dock early because there's a bad outbreak of Norwalk virus. Everybody's got diarrhea and sicker than a dog. Eastern equine encephalitis virus, this is vectored by an insect. A lot of these viruses are. This is same for the Western. Yellow fever, I believe, is vectored by a mosquito. Same for St. Louis encephalitis virus. Rubella, you've got inoculation when you're a kid, right? The rubella virus, rubella vaccination. Dengue fever is a tropical disease caused by a mosquito bite. A lot of these are carried by arthropods or insects. Anyway, Ebola. Heard about that in Africa. Lots of people died about 6-7 years ago of Ebola. Still around, by the way. Mumps and measles. So if you ever want to go back and find out like what virus causes what disease or what illness, and is it a DNA or an RNA virus, and its taxonomy, this is just a nice table that you can reference. So let's talk about how an animal virus, which is going to be an enveloped virus, right? Because most enveloped viruses are animal viruses. How do they make use of a host cell to complete their life cycle? Well, there are several steps. I think we're going to be talking about 6 different steps. Here's the first, referred to as adsorption. Please note the letter D, because I don't want to be reading about absorption being the first phase of animal virus replication, because there isn't any such term related to that. It's adsorption. Adsorption. So the virus adsorbs to the surface of the host cell, utilizing again specific surface molecules that the virus has that literally will fit into special receptors called glycoproteins on the surface of the host cell membrane. Again, don't forget what a plasma membrane is made up of. AP2, check it out, chapter 3, or AP1, chapter 3. And I'll go through these in a diagram with you in just a moment. So the virus adsorbs to the surface of the host cell. The genome, being the DNA or the RNA, enters the host cell. We'll talk about how that can happen in just a few moments, but it's got to get in, penetration. Once inside, we need to uncoat the viral nucleic acid from the capsid. Remember, the capsid is the outer protein surface structure, outer covering. Just underneath that is the nucleic acid, so we have to uncoat the nucleic acid. The host cell then must synthesize new viral components. Now, when I say viral components, what do I mean by that? Well, you have to ask yourself, what is a virus made of? Basically, 2 things. Outer protein coat, inner nucleic acid, core, right? So if you want to make more viral particles, what do you need? More proteins and more nucleic acids. The host cell makes all of that. How? From the instruction booklet that the initial virus carried with it when it entered the cell. The host cell then will assemble these new viral particles. It'll take the nucleic acid, it'll surround it with capsid, and that's the virus. The host cell does all of this. And finally, then the virus has to have some way of getting out of the host cell. And there's a couple of different ways that this can happen. As it mentions there, one is by budding away from the cell in a process called exocytosis, chapter 3, ANP1, or cell lysis. What's that mean? It means the cell ruptures, pops, right? Okay, so here's that diagram that I was alluding to. This is in your book, figure 6.11. And I'd ask you to go back to the preceding slide where we listed all of those 6 different stages or phases of viral replication. Go back, look at that, flip back and forth between this diagram and that table, if you will, or they do a good job describing it here as well. So follow through these different steps. Now, I have a couple of videos here that I am not going to play as part of this lecture. But I do want you to watch those 2 videos, and they'll talk a little bit about this process of how the virus penetrates the host cell. There's a couple different ways that can happen, and this video does a nice job just talking about 2 of them. This National Public Radio video is sort of just a fun, I think it's a 3.5-minute video that kind of gives you an overview of how viruses work. It's just sort of a fun video to watch. So please do watch that. This is something you need to know upside down, left to right, right to left. You're going to see questions on this, I promise you. So know viral replication in animal cells. Know what's going on, be able to explain it. It's not hard. Okay, so a few more specific comments with respect to these different stages, if you will, of viral biology. Going back for a moment to the adsorption step, which is the first, of course, in the sixth, where the virus uses its outer surface molecules to dock with the cell membrane of the host cell. And so here we can see an enveloped coronavirus, i.e. COVID-19, right, or any number of other types of coronaviruses, utilizing their glycoprotein spikes rather to fit into special receptor sites on the cell membrane. Naked viruses, which by definition lack an outer envelope, they don't lack an outer signature, right, chemical signature, as we saw back earlier in the icosahedral type of naked virus. We have these spikes that stick out. They do also have the ability to dock with special receptors here on the surface of the cell membrane. When we talk about host range, this gets back to the idea that only certain kinds of viruses can target certain types of cells. So for example, in your body, a given virus may only be able to dock with and enter a specific cell type. You're made up of billions of different kinds of cells, right? Think of the major types of tissues, for example, epithelial, connective, muscular, nervous. Within that group, there's different subcategories, right? Hepatitis B virus, for example, can only infect liver cells. It will not touch bone cells. It won't bother your skin cells. Your nerve cells will just not let them in. They don't have the proper receptors. Only liver cells have the proper receptors to fit the outer proteins of hepatitis B. In the case of HIV/AIDS virus, there's a certain protein on the surface of helper T cells called the CD4 receptor that the AIDS virus spike fits into. Okay, this virus cannot get into any other cell other than helper T cells. And again, AMP1, lymphatic chapter, we talked about that. This should not be something you've never heard of. Helper T cells are very important in the immune response. They're absolutely critical to the immune response. If you have tons of helper T cells invaded by AIDS virus, they cannot assist in fighting off diseases that you could ordinarily fight off with no trouble. But when an AIDS patient, for example, in late stage AIDS, which we don't see nearly as much as we did 30 years ago when people were dying right and left, right, due to AIDS, but still people die today of it, don't get me wrong, it's still here. It's the opportunistic diseases, we call them, that kill the patient. It's diseases that you and I can fight off, no problem, because our helper T cells are functioning properly. But if you've got AIDS virus in you, and lots of it, and all those viruses are targeting helper T cells, then your helper T cells can help mitigate and orchestrate an immune response to another pathogen. And so that pathogen, that opportunistic pathogen, ends up killing you. So again, back to this idea of host range. Rabies viruses, you may have heard of this before. If an animal is thought to be rabid, and they want to confirm the rabies in the animal, that they send the brain to the lab to be examined, because it's the brain, the nerve cells in the brain, that the rabies virus gets into. Yeah. So you get the idea. Certain kinds of viruses target certain kinds of cells. Once we have the adsorption of the virus. Then, of course, we have to have penetration. And this is what that video that I talked about earlier, the first of the two videos in the preceding slide, talk about fusion, for example, here. Fusion is simply discussing how this enveloped animal virus is coming into contact with the surface of the host cell. Remember, the spikes fitting into the glycoprotein receptors. And in this particular example called fusion, the outer envelope here becomes part of the cell membrane. And it is the nucleocapsid minus its envelope that's actually brought into the cell. Now, eventually, we're going to uncoat that, right, in step 3 or 4, and out pops the nucleic acid. All right, that's called fusion. So the outer cell envelope fuses with the cell membrane. That's basically what that means. The other way that you can have penetration is through the process of endocytosis. And this was described again in that video. In endocytosis, here we have the enveloped virus, or it could even be naked virus, coming into contact with the host cell membrane. And look what happens here. We have the cell membrane invaginate, and we create a vesicle. You've heard about vesicles in A&P. And that vesicle is a membranous outer structure made of cell membrane within which is the enveloped virus. Eventually, the vesicle and the envelope will degrade, and the capsid will break down, and the nucleic acid will be freed into the host cell. In the case of naked viruses, it's also endocytosis. There's still the formation of a vesicle, but it's a little bit different in terms of how the virus interacts with some of these surface structures of the cell membrane. You also notice here that the vesicle doesn't break down, or the nucleocapsid like we saw earlier in the envelope virus. It sort of is the nucleic acid sort of unwinds itself and is released out the capsid and then eventually out the vesicle. That's kind of interesting. So it's a little bit different mechanism. But either way, we got to get the nucleic acid into the cell, right? Because that's what the instruction booklet, right? That the cell is going to be reading to help make more virus. So here we are in that process called synthesis. Now, the way in which a DNA virus does this versus an RNA virus is a little bit different. But you have to have some fundamental knowledge of what transcription and what translation are, those two steps. Normally, if you want to replicate DNA virus, that process occurs within the nucleus of the host cell. That sort of makes sense because that's where the DNA nucleotides are. However, if it's an RNA virus, that process of making more RNA is taking place in the cytoplasm of the host cell. So again, connect DNA viral replication with that taking place in the nucleus, RNA viral replication or assembly, synthesis in the cytoplasm of the host cell. One of the first things that occurs once the host cell, it has utilized the nucleic acid of the virus, it's now reading the instruction booklet that the virus gave it. And on page 462, it tells the host cell how to make the viral protein, the viral capsids, the viral capsomers, which together are assembled into the capsid, right? The outer shell. So we assemble this empty shell first. The host cell does that. Then we have to take the newly synthesized DNA or RNA, and that gets packaged inside, right? And if we're talking about an enveloped virus, before that virus can leave the cell, it has to somehow acquire the envelope with those spikes on it, doesn't it? And a lot of times, enveloped animal viruses acquire those on the surface of the host cell, on the membrane of the host cell. So here we see that process and how it takes place. This is really fascinating. So here's the release of the virus. And so we've replicated the RNA. The cell has done that for us. The cell has also made a capsid. And here's the viral nucleocapsid that the host cell has manufactured. And we have to yet acquire now the outer envelope. And how do we do that? Well, the host cell also makes those viral glycoprotein spikes and puts them on the surface of the cell membrane. So you can see those as purple structures here. And there's probably more than just one type, because remember H2N1 earlier. So there's going to be, in that particular instance, at least 2 different varieties of glycoproteins. So this is just showing one for simplicity's sake. And we have what's called the viral matrix protein here in green. And here's our viral nucleocapsid coming into contact now with the viral matrix protein, and the formation now of what's called a bud, if you will. It's really exocytosis. And here is our recently budded enveloped virus. This is the leftover cell membrane minus the viral matrix proteins. There could still be other viral spikes here, right? Because there may be another viral nucleocapsid just behind this guy that's going to eventually bud off too, because host cells can bud many thousands of viruses, in a very short period of time. This is an amazing photograph. I don't know exactly why they didn't keep this in the book, because I think it's very cool. I think it was one of the earlier editions. So here's our host cell. Here's the nucleus, you can see, and here's the cell membrane in red. And of course, we've got our internal structures. Here's what appears to be some virus forming. And then here's a bud. Here's one that's just getting ready to leave the cell. These have already budded off. And we can see the nucleic acid in yellow. And then here's our envelope sort of shown in red. Budding from the cell. Now, when the cell forms these buds, it's not dying, is it? It's still intact. It's still a viral factory. There are times, though, that the cell will actually lyse. And again, I am not going to play this video. I would ask you to just simply watch it. It's going to go over the budding process.
Enveloped viruses are...
So I'm going to go to the next slide. This describes how a cell lyses when it releases its virus. So oftentimes poxviruses and bacteriophages will, if you remember, those are the complex viruses, will often be released by a cell by inducing lysis of the cell. The same applies for many naked non-enveloped viruses. They often tend to cause the cell to lyse, so the cell dies in the process. Enveloped viruses typically don't cause lysis of the host cell, like we saw in the preceding slide. So how long does it take from the time the virus adsorbs to the surface of the cell membrane to the time that it either buds off the cell or causes the cell to lyse? Well, that's what this kind of gets at. It can be as short as 8 hours in the case of poliovirus, or in the case of many types of complex viruses, it can take 3 or 4 days or more from onset, i.e. adsorption, to the time at which the cell lyses or buds. Just depends on the virus. But it doesn't happen in 10 minutes. This takes hours and hours, sometimes days. So how many virions, which are another term you could use for viruses, how many virions are released from a host cell at one time? Well, hard to imagine this, but as we saw just a few moments ago, some of the complex viruses that can induce lysis of the host cell, like we saw in that earlier slide, can release 3,000 to 4,000 virions. A single cell can do that at one time. And then each one of those virions can go on to infect another host cell, and in a rather short period of time, right, a couple days, they can release 3,000 to 4,000 virions. And if that continues on and on and on, it doesn't take long before you're dealing with exponential numbers here with respect to a viral load, let's say, that a patient might carry. Polio-infected viruses, or cells rather, can release over 100,000 virions at one time. So it doesn't take long for the viral load, we call it, to get to substantial numbers in an individual. It can happen quickly. It all depends upon the replication cycle, the timing. It depends upon the health of the patient. There's a lot of mitigating circumstances that can impact, how an individual might react, let's say, to a certain type of viral infection. But the point is, viruses have amazing abilities to replicate rather quickly and overwhelm a patient's immune response, possibly. And that's what we're seeing in COVID-19. We see people walking into a hospital on Thursday and they're dead on Sunday, right? You've all heard that. That is true. It can happen very fast. Now, again, typically that doesn't happen to a healthy 18-year-old, but immunocompromised people with underlying health conditions, as you've heard, are most susceptible to COVID-19. The last thing that I'm going to leave you with before I end this long lecture is what damage can and do viruses have on host cells. We know that the host cell acts as a factory for the formation of the virus, so it's helping the virus out significantly. But over time, how do some host cells exhibit cellular reactions, if you will, to having been invaded by a virus? And so we're going to talk a little bit about what are called cytopathic effects. So these are visible changes that we can see to a cell or a cluster of cells after they've been invaded by virus. One thing that we can notice is a significant change, not only to the function of the cell, but there's some morphologic, pathogenic changes that we can see exhibited. The cell shape may get bigger. Its overall shape can be altered to the point where we don't even recognize it as a nerve cell or a liver cell or whatever it might be. And oftentimes, cells that have been invaded with virus exhibit what are called inclusion bodies. So in this particular stain, which is not in your book, this is from an earlier edition. And I wish they would have kept it because I do like it. They have a slightly different photograph in your book, but nonetheless, these green structures that you can see are these so-called inclusion bodies, as it indicates here, are often indicating where we have clusters or masses of viruses, or maybe ribosomes that have been damaged due to their role in helping to make more viral proteins or what have you. So cellular organelles can be significantly impacted. They can actually kind of all cluster and merge together to form these so-called inclusion bodies. And the other fascinating thing that we see in host cells is their ability to fuse together and form what we refer to as multinucleated cells that are collectively called syncytia. So here we have what's termed a giant cell, which at one time consists of normal cells that have been infected by virus, and now they kind of all coalesce, their cell membranes fuse together. This is really bizarre, but this is what the cytotechnologist would see under his or her microscope. You'd see these masses of quote-unquote giant cells, which were at one time healthy cells, but now are exhibiting this very kind of strange cytopathology. The cell can lyse, as we know. That's one of the ways in which viruses get out of host cells. The cells die, they pop, they lyse. So that's pretty significant damage to a cell, I'd say. Cell dies in the process. And sometimes we even see the cell's DNA getting impacted and altered as a result of viral infection. And this brings us to an interesting topic that we will spend a few minutes on later when we come back during the next lecture. But when we talk about transformation of cells, sometimes this occurs as a result of viral infection that so alters the cell's DNA that mutations form in that cell and cancer results. Now, again, sorry to harp on this, but go back in ANP1, chapter 2 or 3, I guess it was, on cells. There's a section on cancer. What is cancer? Cancer, remember, is where the cell's regulatory cell division mechanism is screwed up. If you remember, we have green lights and we have red lights within the cell cycle. The green light tells the cell to go on, to proceed from that point into the next stage. Maybe it's mitosis. Maybe it's cytokinesis. Maybe it's entering S phase of interphase, which causes DNA replication. But you got to understand that a healthy normal cell has well-orchestrated cell division mechanisms. It knows when to start replicating. It knows when to stop replicating. In the case of cancer cells, they don't obey those underlying green light, red lights. In fact, there aren't many red lights. They get more green lights. And so the cell continues to divide and divide and divide. And the cells that it produces, they continue to divide and divide and divide. And pretty much you're dealing with an uncontrolled cellular division process, right? Problem. And that can lead to the formation of tumors and so forth. So what's a bit scary is the fact that there are some viruses that you and I can acquire through our everyday living that could, down the road, cause cancer. It's not the food you eat. It's not the water you drink. It's not the genes you inherit from your parents. It's exposure to different kinds of viruses that can cause cancers, different kinds of cancers. Not a ton of them, but there are some. Okay. And I will leave you simply with this table that talks a little bit more about some of the cytopathologic changes that we see in animal cells once they have been infected with certain types of viruses. And this kind of breaks down different types of virus and how certain cells will respond to having been invaded by these different viruses. Again, I'm not asking you to memorize this. I think it's just worth taking a few moments and looking at these and seeing how cells respond. This is all important information to the cytotechnologist or the individual in the laboratory who may be working on a patient that is suffering from a viral infection. He or she may be looking at tissue samples under the microscope and looking for these particular responses in an attempt to figure out what virus that patient might have. Okay, I'm going to stop here and we'll pick up where we left off in the next lecture.